Hello. Today we're going to move on to part 2 of Fundamentals of Pharmacology, Pharmacodynamics. We've spent the last couple of sections discussing all the pharmacokinetic principles. In other words, what the body does to the drug. In the next couple of sections, we'll do the reverse. Pharmacodynamics describes the body's response to a given drug. And in terms of clinical practice, this is the piece that we have greatest interest in. So Section 5, Pharmacodynamics, Introduction to Drug Action. So we're going to return at the beginning of this section to a slide that I have shown you previously. In the paler background in the top half of this particular slide refers to the PK profile. We are now going to discuss pharmacodynamics. This is the effect that one expects to see, or maybe doesn't want to see, i.e. toxicity, in terms of pharmacologic effect. We differentiate between pharmacologic effect and clinical response. Pharmacological effect is the impact of the drug at both the intended and unintended site of action. Typically here, we talk about receptor activity. The clinical response is the sort of response that you as the consumer, or me as the prescriber, might expect to see in response to drug delivery. For example, giving a patient a drug to lower blood pressure, the clinical response would be the blood pressure lowering response. The pharmacologic effect would be the mechanism of action of the drug. How did the drug lower the blood pressure? And you will see at the bottom of this slide, on the left-hand side, that we talk about efficacy, the intended effect, and toxicity, the unintended effect. So when we talk about pharmacodynamics, receptor biology is a cornerstone to our ability to understand drug action. So we will follow in this particular section an introductory to the concept of receptor activation. A subsequent class will talk about the dose response in response to receptor activation. So the learning objectives for Section 5 and 6 include the following. Understand the principle of drug-receptor interactions. Appreciate how receptors determine drug response. Be able at the end of these two classes to define 5 types of receptors. Know what they look like. Know how they work. Know how their function can be modulated. In addition, in these sections, we will spend a lot of time talking about G-protein coupled receptors, one of the 5 type of receptors that I'm going to introduce. Because G-protein coupled receptors form the cornerstone of receptor biology and drug action, we're going to delve into greater detail about this particular type of receptor. So at the end of this, I'd like you to know the structure of a G-protein coupled receptor and the basic sequence of events following an agonist binding to the receptor. I'd ask you to know the 3 classes of G-proteins and the effectors that they regulate. I'd like you to appreciate how G-protein coupled receptor subtypes provide the opportunity of therapeutic leverage. And I want you to learn how receptor activity is modulated. A lot of these terms will be foreign to you. I will introduce them and do my best to explain them to the point where you will be able to understand this material. This material is going to be a bit more complex than what we have thought before. It may require some background reading in addition to what I provide here. I will try to explain these principles as thoroughly as I can. So let's start off with the rudimentary aspects of pharmacodynamic principles. First of all, most drugs bind to a receptor to bring about an effect. The types of actions that we see commonly in human biology and pharmacology are detailed here. The drug, in parenthesis D, plus the receptor, effector, referred to as R, bind together to give you a complex, denoted here, drug-receptor complex. Following creation of this complex, there is a net effect produced, referred to as the pharmacodynamic effect. Another example would be a drug and a receptor, again, create a drug-receptor complex, which elicits an effector response, or the creation of an effector molecule. And it is this molecule that brings about the ultimate pharmacodynamic effect. The third example is the first two steps are exactly the same as the previous example, create the drug-receptor complex. And it is this complex that induces the activation of a coupling molecule to bring about an effector molecule to bring about an effect. So as you can see from these first three examples, we have increasing levels of complexity of different steps that are undergone in order to bring about the pharmacodynamic effects. It is this complexity of drug-receptor interaction that makes pharmacology sometimes challenging to understand when we're evaluating drug responses. The last example is a common example in human biology, whereby inhibition of metabolism of an endogenous activator, this is something that exists in each of us in humans, referred to as endogenous, naturally occurring activator. As a result of decreased breakdown of the natural or endogenous activator, there's increased activator action on the effector molecule, resulting in an increased effect. You could think of an analogy here being just an increased concentration of the endogenous activator. Think about insulin. Think about acetylcholine. These are naturally occurring hormones and neurotransmitters that exist in the body. Under normal circumstances, they're broken down. Any drug that results in the natural inhibition of the breakdown of that endogenously synthesized compound will result in an increased duration of pharmacodynamic effect. So these are four rudimentary examples of how drugs interfacing with receptors bring about their individual responses. What is a receptor? Well, the simplest definition is the following. A receptor is a protein that binds a signaling molecule. We typically in the drug world refer to this as an agonist, and in doing so generates a signal of its own. Let me repeat that. A receptor is a protein that binds a signaling molecule and in doing so generates a signal of its own. Think of dominoes as your analogy. Let's say we have a stack of dominoes lined up on a flat surface. You tip over the first domino. Your tip, your finger, is the agonist. It touches the first domino. Consider that to be your receptor. It causes the falling down of subsequent dominoes. These are the signals that are generated as a result of receptor activation. Receptors themselves are thought to exist in an inactive and an activated form. The inactive form is designated R subcase lowercase I. The activated form of the receptor is R subscript A. Some receptors remain in an active conformation even in the absence of an agonist binding. This is an important concept. It is referred to as constitutive activity. In other words, there exists the phenomenon whereby a receptor can be activated in and of its own right. An agonist is a molecule that binds to and activates a receptor to bring about an effect. I'm going to repeat that because it's a very important definition. An agonist is a molecule that binds to and activates a receptor to bring about its effect. If a drug given in sufficient quantity to saturate the receptor pool binds to the activated form of the receptor and stabilizes it so that a large percentage of the receptor stays in the activated form, it is called a full agonist. In other words, the majority of the receptors that are the natural ligand for that agonist are in the activated form. They are kept in the activated form by the agonist binding to the receptor. This is called a full agonist. Let me show you a picture. I'll ask you to concentrate on the top part of this particular diagram. In the top left corner depicts your inactivated or your inactive form of your receptor. On the bottom part of the left-hand diagram is an inactive form of the receptor with your drug bound to it. On the top right-hand corner is the activated receptor with the drug in equilibrium and the drug binding to the receptor to bring about the activated form of the receptor bound to the drug. You can see here that if you have a drug that binds to a receptor, you bring about a large effect. Now, what about if you have a drug that binds to the activated form and stabilizes it so that a smaller percentage of the activated receptor pool is unaltered no matter what dose of the drug is given? It's called a partial agonist. And the reason for this is that not all of the receptors are maintained in the bound form in an activated state. So if we go back to our picture, here we see a drug in its activated form, partially bound, giving a smaller effect. A third concept that I'd like you to appreciate is an inverse agonist. If a drug has a higher affinity for the inactive form of the receptor and stabilizes a large fraction of the inactive form of the receptor pool, then the constitutive activity that we talked about previously will be lost. So let's say, for example, we have a receptor for acetylcholine. Acetylcholine is a neurotransmitter. It is responsible in part for muscle tone. It is one of the neurotransmitters that allows us to stand for long periods of time through its ability to maintain muscle tone. If we use a drug that prevents the activation of the receptor to convert it from the RI form into the RA form, then we lose some of that constitutive activity. This is called an inverse agonist. It's inverse because it removes some of the natural inherent property of the receptor, the constitutive activity, but it's called an agonist because it's still binding to the receptor. And then lastly is the concept of an antagonist. This is a molecule that prevents activation of the receptor by an agonist. It's different from an inverse agonist for many reasons. One, it binds to a different site than the active site of the receptor. Two, it blocks the binding of the natural agonist to the active form of the receptor, conceptually speaking, like covering it with an envelope. So let's look at our picture again. Down here, we see on the y-axis the response to a drug. On the x-axis is incremental doses. These are shown as log dose response curves because they're easier to interpret than regular dose response curves. So this particular curve here represents a full agonist, a dose response curve following administration of a full agonist. So remember what we said before. The full agonist binds to the active receptor to create an activated receptor drug complex, bringing about the potential of a maximal effect. This is the full agonist dose response curve. This particular curve here refers or represents a partial agonist. Here we see that the drug does indeed bind to the activated form of the receptor, but no matter how much drug you give, shown here along the x-axis of increasing concentration or increasing dose of the drug, the dose response curve plateaus out. You cannot induce a further increase in agonist action. This is referred to as a partial agonist. An antagonist, as we said before, prevents the binding of the natural agonist. So this is a flat line. So the drug response curve represents no activity. And then finally is the inverse agonist. And remembering that the inverse agonist binds to the drug in the inactive form of the receptor. Binds to the receptor in the inactive form of the receptor and eliminates the constituent activity that that receptor would normally demonstrate. So this represents the inverse agonist dose response curve. This appreciation of inactive versus active receptor forms is important in order for you to be able to understand the difference between agonist effect, antagonist effect, and inverse agonist effect. Now, the picture gets a little bit complicated as follows. At the top of this slide, you see what we've talked about before. Drug, receptor, effect. No different than what we've talked about previously. Shown in the top of the slide, panel A, is representative of an agonist binding to the active site of the receptor and activating the receptor to bring about a clinical, a pharmacodynamic plus or minus a clinical response. And that in the dose response curve shown over here on the right is represented here. In addition to this, we have the ability to modulate the response of the agonist, to diminish it. This is referred to as a competitive inhibitor, something that will reduce the maximal response to the natural agonist. And this is referred to in this particular curve here. Finally, we also have the opportunity to incorporate what are called allosteric modulators. These can be activators or inhibitors. In example C, shows the ability of a molecule to bind to a site on the receptor that is not the active site where the agonist binds, but by binding of the allosteric activator, it enhances the response to the natural agonist. So you have the agonist binding to the receptor, activating it, and then you have the allosteric molecule that enhances the response to the agonist. This is shown by this particular curve here. And what this curve demonstrates is that you get a much more amplified response at a lower dose of the agonist when it is present with the allosteric activator. And then finally is the concept of the opposite approach, an allosteric inhibitor. Again, this is a molecule that binds to the receptor, a site that is distinct from where the agonist binds, but by binding the allosteric inhibitor, it diminishes the response to the natural agonist, and that is shown by this particular response here. In these dose response curves, Anything that shifts the curve to the left enhances the overall activity of the drug. Things that shift the curve to the right reduce the activity of the drug or the agonist. So these concepts of agonist, partial agonist, inverse agonist, antagonist, competitive inhibitor, allosteric activator, allosteric inhibitor are all common concepts that allow us to develop drugs, evaluate drugs, and appreciate their potential impact in clinical medicine. One example where we are seeing a huge explosion in the arena of cancer chemotherapy is the concept of allosteric activators, that we are able to use small molecules to amplify the response of other chemotherapeutic drugs in such a way that it permits us to use lower doses of the agonist, thereby possibly avoiding toxicity associated with higher doses of the agonist because we're able to maximally activate the response to the receptor. So allosteric activators is a common approach in trying to amplify efficacy and reduce toxicity, which is incredibly important in cancer chemotherapy. So in this particular section, we are going to talk about receptor activation. And a lot of what I'm going to talk about has relevance to normal physiology and human biology. But my emphasis is going to center on drug-receptor interactions. In order to appreciate this part of the pharmacodynamics section, I want to explain to you some important general concepts. Cells contain a large array of receptors for naturally occurring substances such as hormones, insulin, thyroxine, cortisol, neurotransmitters such as acetylcholine, adrenaline, noradrenaline, or sensory inputs, light, noise, taste, smell. Receptors are activated by an array of endogenous compounds, examples that I just gave you. More than 1,000 different receptors have been identified in the human genome. This is a very large concept to appreciate. The vast majority of all the drugs that we use in clinical medicine today operate through receptors. This includes receptors on enzymes, receptors on cell membranes, receptors within cells, etc. Receptors, importantly, mediate the actions of both pharmacological agonists and antagonists. Receptors largely determine the quantitative relationship between dose or concentration of the drug and the pharmacologic effect. This is referred to as the dose-response relationship. We will return to this concept in the next section in this course. The concept of quantitative dose response is critically important in drug evaluation, both in the development stage of novel therapeutics, but also in the practice of medicine in prescribing and estimating magnitude of response, both effective and toxic. Receptors, very, very importantly, are responsible for the selectivity of drug action, and this I will spend a good deal of time talking about. For most drugs, without a knowledge of the receptors to which they bind, it is very difficult to understand the mechanism of action of the drug, the selectivity of the drug or the lack thereof, and to appreciate the true intricacies of the dose-response relationship. Having said that, there are many drugs that we use in clinical medicine today that have been around for many, many decades in clinical practice, for which we don't know the receptor. But in this era, our development of drugs formally involves an evaluation of mechanism of drug action in an honest attempt to try to appreciate the receptor that mediates the response of any novel therapeutic that will come to market. Most receptors are proteins, and thus they abide by all of the properties that are detailed for proteins in terms of synthesis, biotransformation, metabolism, etc. The best characterized drug receptors are regulatory proteins. They mediate the actions of endogenous chemical signals, such as neurotransmitters and hormones, as I've described previously. Other drug receptors include enzymes, transport proteins, and structural proteins. So the concept of receptor biology is diverse. It affects every physiologic process in the body, but for our purposes, we're going to talk about it in relation to drug action. And specifically, this next section is going to focus on signaling mechanisms and their interplay in bringing about pharmacodynamic drug effects. So this is an overview of agonist action, and I'm going to confine many of my comments here to the effect of agonists, both naturally occurring and exogenally administered in the form of drug therapy. So on the left-hand side of this slide is a list of different types of agonists. First group, circulating hormones, insulin, thyroxine, growth hormone, etc. The second, neurotransmitters, adrenaline, noradrenaline, acetylcholine, serotonin, GABA, dopamine. Sensory stimuli, as I've described previously, light, taste, noise, smell. Autocrine and paracrine factors. These are small molecules that are synthesized within a cell and activate receptors on their own cells. These are autocrine, or activate neighboring cells, paracrine. Paracrine, an example, would be prostaglandins, small proteins that bring about a myriad of effect. Cytokines can be examples of autocrine and paracrine factors that function as agonists. And then finally, the focus of today's discussion, therapeutic agents. Therapeutic agents, for the most part, can be synthesized to mirror endogenously synthesized compounds. For example, we give insulin to patients with type 1 diabetes. The insulin that we give patients is synthesized based on our knowledge of the naturally occurring hormone. Adrenaline, we give this to patients also, and it is synthesized based on our knowledge of the naturally occurring neurotransmitter adrenaline, also referred to as epinephrine. And then we have therapeutic agents that are completely synthetic agonists that have no resemblance to any naturally occurring endogenous compound. So this is our array of agonists. They bind, as shown here, either on the cell membrane to a receptor. They can also cross into the cytoplasm and bind to an intracellular receptor shown here. There then ensues what is referred to as a proximal intracellular effect. And this is the involvement of a whole series of what are referred to as second messengers. The synthesis of these second messengers causes a cascade of the synthesis of a series of enzymes that ultimately will result in a pharmacodynamic effect. In addition, you can see down here the concept of the recruitment of adapters, which may function as an allosteric activator, inhibitor, or just as an amplifier or a reducer of the proximal intracellular effect. So agonist, binding to receptor, bringing about a series of second messenger signaling pathways. Excuse me. So what are these receptors? Well, there are essentially 5 distinct types of receptors and signaling mechanisms involved in agonist and response relationships. The first of these, a lipid-soluble chemical signal, crosses the plasma membrane and acts on an intracellular receptor. 2 and 3 involve a signal binding to the extracellular domain of a receptor, causing an intracellular activation of signaling pathways. Number 4, it's where a signal binds and directly regulates a channel on the surface of a cell membrane. And then finally, a signal binds to a cell surface receptor, which is a G-coupled protein. We're going to delve into each of these and give you examples from human biology. This is a picture of the 5 different signaling pathways. The first example showing you the intracellular receptor. The second and the third example showing you the extracellular domains with the agonist binding, bringing about intracellular second messenger signaling pathways. The third showing you an ion channel that actually sits across the membrane of the cell and regulates the passage of ions. And then the final example, G protein coupled receptors, where you have an extracellular domain and an intracellular domain linked to a G protein. So first of all, starting out with the first example and remembering what we said before, the first example is a lipid-soluble chemical signal, crosses a plasma membrane, acts on an intracellular receptor to bring about its effect. The best example of this is corticosteroids, cortisol. So let me walk you through this schema. This represents the cell. Everything inside. This represents the cell membrane. Outside of the cell is circulating plasma. So here you see the agonist, steroid, corticosteroid in this example, being delivered to the cell membrane. Just to bring you back to the pharmacodynamics section of this course, it is bound to plasma protein. In this particular example, it's corticosteroid binding globulin. This is the plasma protein that specifically carries corticosteroids to the cell surface in order to mediate its pharmacodynamic effect. So the steroid is delivered to the cell membrane. It crosses through the plasma membrane. It is lipid soluble. It enters into the cytoplasm. When it enters into the cytoplasm, it seeks out the corticosteroid receptor, here designated as R. Now, in the natural inactivated form of the receptor, as we referred to previously, there is no steroid bound to the receptor. However, the receptor is kept in what is referred to as a ligand-friendly state. It is kept in this conformation by heat shock proteins. These are small molecules that exist commonly within cells. Upon exposure to the steroid, which has just entered into the cytoplasm, the receptor becomes activated. The steroid binds to it, and binding to the receptor activates the receptor, and the receptor loses the heat shock proteins as shown in this part of the diagram. So you now have your steroid bound to your receptor, your steroid receptor complex, or your drug receptor complex. This has no effect in the cytoplasm. It has to cross through the nuclear membrane shown here. The steroid receptor complex enters into the nucleus, and it seeks out a particular part of the DNA strand called the glucocorticoid response element, GRE. Once the steroid-bound receptor binds to the glucocorticoid response element in the target gene, there is a turning on or turning off of transcription. It either increases the synthesis of proteins or decreases the synthesis of proteins depending on the cell and the particular necessary response. This is an example of intracellular receptors for lipid-soluble ligands, steroid, glucocorticoid, steroid hormone receptors. The onset of action typically takes about 30 minutes to several hours. So what we are seeing here is the process involving multiple different steps, ultimately resulting in an altering in the synthesis of proteins. The duration of response can last for several days because of the relative slow turnover of enzymes and proteins. So once you turn on these receptors, it is expected that the pharmacodynamic effect of turning on the receptor is going to have a reasonably long duration of action. We use this information clinically. It is very important how we use this information clinically, and this is an aspect that you would learn in future courses. The second receptor that we're going to discuss is where the signal, the agonist, binds to an extracellular domain of the receptor, activating the enzymatic activity of the cytoplasmic domain of the receptor. So the example that we're going to use here are cytokine receptors. So the structure of cytokine receptors, and I'll show you a picture in a moment, is such that they have an extracellular and an intracellular domain. They span the cell membrane. The intracellular domains of these receptors exist as monomers. However, when the agonist binds to the extracellular domain, they dimerize. They come together to form couplets. So the signal binds to the extracellular domain of the receptor, activating the enzymatic activity of the cytoplasmic domain via dimerization. These receptors cannot be activated, insofar as we know, without dimerization. Once dimerization occurs, separate mobile protein tyrosine kinase molecules, referred to as JAK, are activated, resulting in a series of phosphorylation of signal transducers and activation of transcription molecules, referred to as STAT molecules. Again here, much like the intracellular receptor pathway, we see multiple steps that are necessary to result in a pharmacodynamic effect. The STAT molecules ultimately travel to the nucleus, where they regulate transcription. So unlike the previous example where the ligand-bound receptor travels to the nucleus, here it is the product of the activation of the receptor on the cell membrane that is transmitted into the nucleus to activate transcription. Common ligands in human biology for cytokine receptors include growth hormone, erythropoietin. This is a hormone that regulates the synthesis of red blood cells. Interferon, also a naturally occurring cytokine that is a regulatory component in controlling inflammatory processes. So let's look at a picture to try and explain this a little bit better. This is your cell membrane. Here you can see the extracellular domain and the intracellular domain. In the inactive state, these receptors exist as monomers. Along comes a cytokine molecule. Remember, cytokines from inflammation, they contribute to normal inflammatory and also deranged inflammatory responses. When the cytokine molecule binds to the receptor, the cytokine receptor, you get dimerization of the receptor and activation of this series of second messenger signaling pathways, ultimate effect being the creation of STAT molecules that cross into the nucleus and alter transcription of proteins. This particular example is very important in cancer biology. We know that cytokine molecules can contribute to the growth of certain tumors. Inhibitors of cytokine receptors are currently being evaluated for their potential utility in a wide range of different cancers. The third example is where a signal, again, is binding to the extracellular domain of the receptor, but in this case, it's bound to a tyrosine kinase, which it activates. These, not too surprisingly, are referred to as receptor tyrosine kinase. Again, another huge area of drug discovery for cancer chemotherapy. So to describe this in the same manner as I described the previous two types of receptor, this receptor has an extracellular hormone binding domain, a cytoplasmic enzymatic domain, typically a tyrosine kinase. So here the tyrosine kinase is a component of the receptor itself. In the cytokine receptor example, the tyrosine kinase was recruited after receptor activation, so an important distinction. The signal, in the example of receptor tyrosine kinase, the signal, typically a hormone or a growth factor, binds again to the extracellular domain of the receptor and it activates the enzyme. Upon the ligand binding, and I'll show you a picture in a second, the receptor converts from its inactive monomeric state to a dimeric state, exactly the same steps as the cytokine receptors, in which two polypeptides, receptor polypeptides, bind non-covalently. The cytoplasmic domains in this example become phosphorylated on the specific tyrosine residue, and their enzymatic activities are activated as a result of phosphorylation. This ultimately results in phosphorylation of a series of substrate proteins to bring about the ultimate response. Natural endogenous ligands for receptor tyrosine kinase include epidermal growth factor and insulin. As I said previously, inhibitors of receptor tyrosine kinase are becoming widely used and are under further evaluation in the setting of cancer chemotherapy. So here's a picture, and you can appreciate that on the left-hand side, this looks somewhat similar to the cytokine receptor. The difference, of course, here is you have your ligand binding to the extracellular domain, but upon binding, you see here that the binding results in dimerization, but here you can see the example of phosphorylation occurring, ultimately of the tyrosine residue, to bring about a second messenger signaling pathway. The fourth example is that the signal binds and directly regulates the opening of an ion channel. So I'm sitting here talking to you today. I'm hoping that you're listening. In order for you to be able to listen, your autonomic nervous system needs to be in top action. You're concentrating, at least I hope you're concentrating. The receptors that are involved in mediating your ability to try and concentrate include ion channels. So let's talk about this fourth type of receptor. So these are commonly referred to as ligand and voltage-gated ion channels. Many useful drugs in clinical medicine today work by mimicking or even blocking the action of natural ligands that regulate the flow of ions through plasma membrane channels. Those of you who have taken physiology before appreciate that action potentials across membranes is particularly important in neurotransmission. The ability for sodium, potassium, and calcium as just a few examples of naturally occurring ions in vivo are necessary for normal neurotransmission. Ion channels are typically composed of several subunits. The ligand, again, in this example, binds to an extracellular domain on one of these subunits of the receptor. And when that ligand binds to a subunit in the ion channel receptor, it opens up a central transmembrane, typically aqueous ion channel, and allows the passage of the ion into the cytoplasm. Again, common examples are the neurotransmitters serotonin, acetylcholine, and glutamate. So let's look at a picture. So in this part of the diagram, we have essentially sliced open the receptor in a longitudinal fashion. And you can see in this particular example 4 subunits. There are in fact 5 subunits for this particular receptor, but we've removed the one most proximal to us. So here we can see a depiction of 4 different subunits, each with Greek suffix lettering. This is an example of the nicotinic receptor. Nicotinic receptors mediate the effect of the neurotransmitter acetylcholine. And you can see that this receptor spans the plasma membrane. It literally sits on the plasma membrane. And what we are attempting to show you in here is that this is where the passage of the ions occur. So the ions are either moving from an intracellular to an extracellular location or vice versa. And it is the regulation of this aqueous pore down the center of this channel that permits or inhibits the flow of enzymes from the flow of ions from the extracellular to the intracellular domain. It might help you by looking at this particular graphic insert on this slide. This is now looking at the receptor from above, so looking down into the receptor. This section here is the aqueous pore through which ion transport needs to occur. And the aqueous pore is surrounded by these 5 subunits as shown for the example of the nicotinic receptor. So binding to this receptor, acetylcholine in this example, binding to the alpha subunit of this receptor opens up the ion channel and allows the transport, in this example, of sodium ions. But we have many drugs that actually function as inhibitors that prevent the opening of the ion channel and thus prevent the transmission of ions, resulting in many cases in hyperpolarization of cell membranes and diminished pharmacodynamic responses as a result of the agonist binding to the receptor. So far we've talked about 4 transmembrane signaling mechanisms. We're going to end there for today's class. And in the next section, section number 7, we will introduce G-coupled protein receptors and do an in-depth discussion of those. Thanks for attending class today.